Hello, everyone, and welcome to today's webinar titled Innovative Drug Development at a Glance, the Concepts, the Vision, and the Factors to Consider. We are very excited as this is the first in a new introductory webinar series on complex innovative trial designs. I am pleased to introduce our presenter today, Zoran Antonijevic. Zoran is Head of Biometrics at MedSource. He has held executive positions in pharmaceutical companies and CROs and has designed more than 100 clinical trials in numerous therapeutic areas, many of which included adaptive designs. Zoran is a longtime chair and leader of the DIA Adaptive Design Scientific Working Group. He has authored numerous papers and scientific presentations and was editor of books, Optimization of Pharmaceutical R&D Programs and Portfolios, and Platform Trials in Drug Development. It is now my pleasure to turn it over to Zoran.
Thank you very much, Alisa. So as Alisa explained, this is the first one in the series of webinars on complex innovative trial designs. And this one will actually set the stage for the upcoming presentations. So this is what I will be covering today. Essentially, as you see in the first three lines there, I will go over some reasons and some reasons to incorporate complex innovative designs in your programs, and go over some advantages and values that these designs bring to drug development. I will then go in pharmaceutical frameworks, which is really I will put these designs in a larger context and how they fit into development programs and portfolios. And I will actually spend a fair amount of time on that part of the presentation. Finally, because COVID-19 is actually involving many of these innovative designs, and already existing trials are very much all innovative designs, I will just spend some time on explaining how innovative designs really fit into development of treatments for COVID-19. This is by no means a presentation on COVID-19, but I think it's inevitable that I spend some time in the end and explain, how innovative designs can accelerate and improve COVID-19 therapy development. So first, I will set the stage and kind of answer some questions. Why complex innovative designs? The focus will be on adaptive designs and master protocols. So here are some of the key reasons why we need to consider changing the paradigm in drug development. And for the large part, we have already done that, and we have seen changes, but there's still a lot more to come. So always the first and foremost the patients, and nowadays patients are often waiting for therapies longer than they should. And even more problematically, sometimes they receive suboptimal therapies. We know that billions have been wasted in drug delays in bringing drugs to the market, and even maybe more so in spending and investing in unproven treatments and failed products. And finally, in many occasions, promising programs get unfounded because budgets are necessarily large. And I will be talking a lot about how to optimize budgets and how to optimize drug development. So that goes a little bit beyond just these innovative designs. It does go into the category of the context. How do you implement these designs in your portfolio? A hole-in-one is highly unlikely even for the pros. So now it's a good time to define the adaptive design. And at the end of this slide, I will define the master protocol as well. So an adaptive design is a design that allows you to change major parameters of your trial based on the data that was observed within that trial. And all the criteria for changing these parameters has to be pre-specified. The clear decision criteria pre-specified prior to initiation of the trial. It is implemented at an interim analysis, usually by an independent board. In phase 3, it has to be implemented by an independent board, but in phases 1 and 2, it's still preferable to for these changes to be implemented by a data monitoring committee. Some of the key parameters that we adjust are based on uncertainties related to treatment effect, to inclusion of subpopulations or focusing on subpopulations, and the dose selection. The main problem with so-called traditional designs that didn't have that flexibility to change things in the middle of the trial were that we come to phase 2 or phase 3 with limited information, and oftentimes we over-engineer designs. Particularly, we have too large of the sample size. We are too conservative with our assumptions, and that increases the cost of trials. And I will later explain how that impacts portfolios. Or on the other hand, not being afraid of overspending, having too small sample size, not investigating enough, and treatments failing when they shouldn't. There's so many situations where actually potentially good treatments have failed because of suboptimal strategies. So complex innovative designs enable researchers to mitigate uncertainty by pre-planning ways to adapt. Before I go into the next slide, I will also define master protocols. Master protocols are a little more recent concept. They have been incorporated into drug development a little more recently, and they're actually addressing this problem of us doing one trial for one indication for one therapy, and just doing things on one-by-one basis. And obviously that's very tedious and very ineffective. And people have come to the realization that we can start working on trials that may include multiple therapies for one indication, or include or study multiple indications on one drug, and improve the efficiency of drug development. And of course, why not? We could have a whole matrix of multiple therapies and multiple indications. And I will go into more detail with all this, but that is what master protocols are. And I will, of course, just like with adaptive design, explain all the advantages related to application of master protocols. Sorry, this is a little slow. Okay, so in general, at a high level, what are advantages and disadvantages of adaptive design? Adaptive designs and master protocols can reduce cost and time of development, and they can improve probability of success. Now, reducing cost and time of development usually comes together. Improving probability of success, usually you cannot accomplish with simultaneously reducing cost and time of development. So it's usually either or. However, in most cases, as it has been demonstrated, these innovative designs can also improve the expected net present value, which kind of incorporates cost, time, and probability of success all at once. Negatives of incorporating innovative designs, and I will be a little bit biased here, I will admit that, they require more upfront discussion. It's a time wasted in terms of drug development, but nevertheless, time spent on upfront discussion is very well spent time, and I think it's almost necessary. And again, another thing that I will cover in more detail. Operationalize the process among multiple parties. Again, there's really no disadvantage in having harmonious process that incorporates multiple parties and improves the communication, but it's something to be really worked upon prior to initiating the trial. And increased statistical resource at planning stage, such as simulations. Again, this shouldn't be a new statistical resource, and I know I said this many times, it's really the statistical resource that nowadays, and particularly in history, spending the time on tables, listings, and figures, where that statistical resource is really not that necessary, and moving statisticians to the aspects of clinical planning and clinical development that's more relevant to the field. And finally, with some of these adaptive designs, there is an increased spend on manufacturing costs and drug supply. If you look at the balance of increased cost on manufacturing versus the improvement in expected with implementation of adaptive designs, the spend is usually dwarfed, but it's something that has to be acknowledged, and I will point out to design that is particularly prone to additional spending on manufacturing and supply costs. This is an overview of advantages of various adaptive designs. You can just take a quick look now. I don't want to spend time on this slide now because I will be addressing this in more detail during the presentation. So now it's the time for a poll. So it would be great if everyone can just answer this question, and we will get results at the end of this presentation, possibly sent by email to all participants. So I will give you a few seconds to respond to this, and then continue with presentation. Okay, so the term complex innovative trial designs is really the result of the newest FDA guidance on this topic, and it's been the draft has just been published in 2019, and many of you may be familiar, but we had regulatory guidance on innovative designs for a long time now. The very first one by EMA back in 2006, and then an FDA CBER guidance in 2010, one of the most anticipated guidances in the history that I remember. That was really highly anticipated guidance. This was followed by CDRH guidance on adaptive design in 2015. And then a completely revised draft by CDER on adaptive design in 2019, and draft guidance on master protocols in 2019, master protocols in oncology specifically. There's another relevant guidance, and that is the guidance on Bayesian statistics in 2010. So why did we need another guidance? Well, first of all, the focus of this guidance on the complex innovative design is on the interaction between the FDA and the sponsor. And I will just, everybody should read the guidance. There's really no need to go over every detail of that guidance, but just a few points. The primary focus is on so-called confirmatory trial, or trial that intended to bring the confirmatory evidence, phase 3 in particular. The greatest focus is on designs that require simulations in order to demonstrate or describe the operating characteristics of the trial, and in particular the control of type 1 error. This was a very strong point in the 2010 guidance, and has been a topic of a discussion over the years. So that is one of the things that most of these trials will actually require simulations that are submitted for the review of complex innovative design. But what I like the most about this guidance is exactly what I will be spending a lot of time on today, and that is the context. So the fact that every design may not be appropriate in every situation, and sponsors, when they're submitting these designs for review, they need to specify the reasons the design is being proposed. They need to specify the setting and validity of that design in particular setting. And it is important that to be understood that what may be acceptable for one indication may not be acceptable for another, and what's not acceptable for one may be acceptable for the other. So it's really focusing, asking people to focus on the context. Okay, this slide I can spend a whole hour talking about, and this is really an illustration of what I will be covering today. So just a few words. Traditional design is what we are referring here to. Traditional design is design that doesn't have any interim analysis, and it's not addressed in a bigger context. It's an individual trial without any flexibility. It was actually the standard way of running clinical trials once upon a time. Some of you may not remember that, but I remember those days. And because of the needs of the industry that I will not go over now, but whoever is interested to ask the question in the end, I can go over the reasons why that might have been acceptable and working back 20 years ago, but not any longer. Now, the first realization is that we need more flexibility in our designs, therefore adaptive designs. The next realization was that, going one trial at a time for each indication and each product is just not sustainable, particularly at early stage in development, particularly in oncology, where we now have realization of how many different indications we have. A company, mid-sized company, cannot afford in any sense financially or logistically to go on a trial by trial basis. Master protocol is really becoming a necessity. Program optimization is kind of a different branch from master protocol. It's not necessarily following master protocol, but it's realization that you cannot be making decision on any individual trial in isolation, that we should be making the decisions at the program level. And I will be covering all this in more detail. And finally, everything just kind of meets together down in the end at portfolio optimization. That's the ultimate level that we should our drug development, every company of any size. Bayesian statistics and real-world evidence are coming more and more strongly into drug development and will become major component of each of these approaches, or each of these designs and approaches that you're seeing here. I know that many people that are attending this presentation are not statisticians, so I will explain what and why a Bayesian approach is. Bayesian statistic is a concept where you start with some prior knowledge and you quantify that prior knowledge in some way. But then as you learn, as you collect more data, you're combining the data which at each stage with the prior, and that prior becomes a posterior. So you keep on updating your knowledge, but now with more and more objective. So you start, potentially you start with the soft knowledge. You may have some hard data for your prior. But then, the data that you're collecting is gradually overwriting by the mean of posteriors the prior knowledge. Obviously, Bayesian thinking is naturally following the process of our thinking about drug development. In fact, it's following the process of our learning about anything. So that's one big reason why Bayesian statistics and Bayesian methods are becoming more popular now. Another one is that output for these for Bayesian statistics is probabilistic. It's probability. It's understood by everybody. It's not a p-value that most people don't understand, and actually even larger number of things that they understand but they don't. It's you're answering the question of interest. Let's say you want to know what's the probability that your treatment is going to provide 30% better survival than what is currently the standard on the market. And as you're collecting more and more data, that's slightly changing, but at each point you're getting that probability. So that really allows you to make decisions in a very clear way. You can even feed them into decision trees. They just naturally fit into decision trees. So that is the reason why Bayesian statistics is becoming more and more popular nowadays. Now we'll go into adaptive designs and types of adaptive designs. Let me say that each and every design that you see here will be covered in a separate presentation by Cytel that's following my presentation. So I'm not going to spend very much time on these designs. This is really just setting the stage and understanding what is out there. So firstly, adaptive designs in phase one, and these are designs that are mostly applied to oncology but could be applied anywhere. The best known one is the continuous reassessment method. It's a Bayesian type of design. Just like many other designs, it has its advantages over the standard 3+3, the old standard 3+3 design. Two key advantages are that you can actually specify the toxicity level of interest, because every indication may or should have the different toxicity level. So you can pre-specify the toxicity level of interest for a particular indication. That's a characteristic of many methods, but not for 3+3. And secondly, as you can see even from this graph, it looks at the entire dose response curve. So so the decision is based on the whole response, model-based response. With 3+3, you're just looking at the current level and making decision based on that. Everything else is forgotten. So that's the rationale why to use this. How does it use? It uses Bayesian principle. This is just a rough illustration. You start with a prior belief of what the dose response may be. You're collecting some data. Let's say at the beginning you're not seeing any toxicity, so you're slightly adjusting your dose response. And then, at the later stages, you may start seeing some, like all these red dots. You'll be seeing more and more toxicities. But after each reading, you keep on revising your knowledge, and you're making decisions about the next dose based on the entirety of the data. And up until you come to the final model, the sample size for these methods is usually pre-specified, and the operating characteristics are determined by simulations prior to initiating the trial. So that was the example from phase 1. Phase 2, adaptive design. This was something that was completely changing the paradigm of drug development. This is more applicable actually to non-oncology. I have rarely seen these type of adaptive dose finding trials in oncology, but that is not to say they're not applicable at all. So the standard way of running these trials was to run with, control and maybe 2 doses, in phase 2, and you decide which one is better. And if you look at the graph on the right hand side, you will see that, well, if you have selected only 2, here it's 10, that may be too many doses. But if you started with only 2 doses, you may be because you come to phase 2 with very limited information. You may be actually on the left hand side end. You may be on the wasted side, and you conclude the drug is not working when it actually is. Or you may be on the right hand side where you may have, for example, too much toxicity. The whole concept of adaptive dose finding studies is that you start with larger number of doses, you have frequent interim analysis, and after each interim analysis, you can reassign the randomization ratio for the next cohort of patients, meaning that you assign more and more patients to doses that look more promising based on the data that you observed thus far. So this is a typical Bayesian concept, and the whole goal is to actually focus more and more on doses of interest, which in this case would be 4 doses in the middle. Any simulations, most simulations that people have been running have demonstrated that these designs are more efficient than standard designs. And the efficiency of phase 2, and I will come back to this point later again, the efficiency of phase 2 actually should be measured with whether you selected the best dose for the phase 3. So group sequential design is the most traditional adaptive design. My guess is that just about everybody on this call has heard about group sequential designs. The goal of group sequential design is to address the uncertainty about treatment effect coming into phase 3. And particularly for cardiovascular outcome trials or oncology trials, you have very little information. You don't know that much about what to expect. And these are the largest trials that you can have, hundreds of millions of dollars, phase 3 oncology and cardiovascular outcome trials. So you need to de-risk it. So how do you de-risk it? You plan your adaptive design up in the most conservative way to the lowest meaningful treatment effect, or the smallest treatment effect that you can afford to study. But then you give yourself a chance to stop early given the data, either for futility or efficacy. And in something like cardiovascular outcome trials, you should have definitely more than 1 interim analysis. So that's the simplest way and most traditional way to address the uncertainty of treatment effect coming into phase 3. Very common design, as I said. Adaptive sample size reassessment is more and more becoming a business as usual, thanks largely to Cytel, but some other authors as well. This has become a very common design. It does address the same question, uncertainty about sample size, just like group sequential design, but you start with more optimistic assumption for the sample size, which is your planned end here. But if the data at the interim analysis seems to be promising, but not satisfactorily good, so I will be covering all the zones here. But let me step back. So let's say that you're planning your study. You're planning with 90% power, 90% of chance, and you're running your interim analysis, and you may have options like in group sequential design for futility and efficacy. You don't have to. But if you look at the probability of that trial being successful, and that probability is greater than 90%, you don't need to adjust anything. You're in the favorable zone, and you just continue the trial as planned. If the probability is less than, say, 50%, then your chances of increasing the sample size of achieving the success are, well, substantially smaller. And also, you need to increase the sample size by a substantial and reasonable amount. So your preference is not to increase the sample size in this zone. However, if data is trending towards the efficacy, then you're tempted to increase the sample size. Then you're in a promising zone. Then you're increasing the sample size because the results are looking promising. But you want to have a greater level of assurance that you are going to succeed. So you want to move yourself from, let's say, 55% back to 90%. That's the whole idea behind this zone, behind this design. This design tends to be slightly more efficient to comparable group sequential design. How do you compare that? If there's a question, I will answer that. It will take me a little longer to do that. But the intuitiveness of the design, that you increase the sample size only when the data is promising, gives this design the additional appeal. In any case, my advice would always be check both and see what works better for you. Run simulations and decide what works better for you. Now, another dilemma that you may have during drug development is whether you want to go with enrichment, whether you want to focus on certain subpopulation, either biomarker-based subpopulation or subpopulation based on some other characteristics in which you may have a better response. This would be particularly common in oncology, but it's not to say that it's not applicable to just about any therapeutic area. So if you go through phase 1 and phase 2, and now you're having some indications that, there is a subpopulation, you're doing your exploratory work, and there's a subpopulation that may be doing better, but you're not convinced in that. So you're not subsetting, your protocol and your enrollment criteria to that subpopulation yet. Instead, you're giving yourself another chance to decide in your phase 3 trial during the interim analysis in the phase 3 trial whether you should be subsetting for this subpopulation or not. And you can craft the decision criteria in many different ways. But for example, just for example, you can say, well, given the data in the interim analysis, if probability of success in this subpopulation is 20% greater than for the whole population, then I'm not going to be enrolling more into patients other than in this subpopulation. And then you continue enrolling only in that subpopulation of interest, and you increase the sample size as needed because now you have fewer patients, obviously, in that subpopulation. So it's another important adaptive design that's really there to help you decide if you should be targeting certain subpopulations and help your drug development and patients as well at the same time. Seamless phase 2-3 trial is another trial. It's not a very commonly used design, but we've seen it a lot, maybe. The advantage of this design is if you look at the traditional design, you would go with control in a number of doses, and then At the end of phase 2, you decide which dose is the best, and then you go into phase 3 and you go with the selected dose and with the control. But now you need to enroll all the patients from the beginning in this separate phase 3 trial. And there are situations when actually you can skip that step, and that's particular when the dose selection criteria can be relatively straightforward. Instead of that, you start with 4 doses. You have an interim analysis for which you usually have a DSMB make a decision, but the DSMB receives the algorithm from you. And at that interim analysis, you select the best dose based on the criteria and the control. The control has to stay there, and then you just keep on enrolling in the control and in the selected dose. So obviously, you're saving a lot in terms of sample size. These numbers are referring to a very specific trial. I will mention that later. But even more importantly, you're saving in terms of development time. You're saving a lot in terms of development time. So the particular method that is mentioned, this is really for statisticians. This was the Advent trial. It was for patients with HIV-induced diarrhea, and the reason that actually this was early on, with implementation of these adaptive designs, the FDA was in favor of this, partly because of strong pressure from the community. The community really needed the treatment, the sooner the better, of course. So as I said, there was a huge amount, there is about 30% in terms of cost and savings in cost and number of patients, but also patients receive the treatment in much more efficient way and much faster without compromising anything from the scientific standpoint. Let's go into master protocols. So as I said, master protocols are protocols where you're studying multiple indications and or treatments in one trial. Why would you do this? There are 3 sets of benefits from implementing a protocol. You have operational efficiency. So you write one master protocol and you may have just one section for each specific indication, but everything else is central and it's standard. So from operational standpoint, and this was probably the first reason that we got into master protocols. Well, you have central laboratories, reading centers are shared. You have a lot of efficiency and standardization from that standpoint. You have common screening platforms. This is a huge issue, and you have common investigators training. Finally, you have consistency of databases. And particularly when you get to platform trials that I will go into later, this gives you a huge leverage. Statistical efficiencies, you have shared control. You can borrow. You need to be careful about this, but you can borrow information among different arms. And as previously said, adaptive randomization can be applied to accrue patients to best treatments. Strategic benefits are extremely important to me. For platform trials, these are perpetual trials. You can keep on adding new trials as the situation allows you, or as they come. You can be adding additional arms. Standardization is implemented at the platform level, and a very important thing is that decisions are made in the broader context. It's not in isolation of one individual trial. There are 2 types of master protocols, umbrella and basket, and I will kind of just skip through this. It's rather obvious. Umbrella, multiple therapies, one product. Basket, one therapy, multiple indications. And this is really just pretty much just explaining the same thing. Umbrella designs are usually done at the exploratory stage of development, and you generally, the drugs, you're seeing the extension, they proceed into the next stage of development. Similarly, the case with basket trials, except so you have some indications proceeding into the next stage of development. Although for basket trials now there is methodology where actually you can implement them in the phase 3 with a strong control of type 1 error if needed. For basket, so it's a matter of negotiating with the FDA if the treatment is working at the indication level or it's really working at the signature level, and all indications or most indications would be benefiting at the same time. MAMS, multi-arm, multi-stage, is actually module is the method, but it's also a module in Cytel Software East, and it's a methodology that actually allows you to do umbrella or basket trials with a strong control of type 1 error. So these treatments that you see, T1, T2, TK, they can be either different indications or different treatments. They can even be different doses, and you have decision points after each interim analysis. This is just an advertisement for the book for platform trials in drug development. It actually covers some other methods for basket trials at confirmatory stage, but it's really a very comprehensive book that's not addressing only statistical methods, but also have inputs from many different thought leaders with different backgrounds and their take on adaptive on platform trials. Okay. Pharmaceutical frameworks. So now I will try to put all this into a broader context of drug development. So firstly, how do you create a framework, and how you should be planning your drug development anyway? I'm starting with this point of perceived risk versus objective risk, and people are reluctant to include innovative methodologies because they think that new things are risky, innovative things are risky, when in reality, in most situations, not innovating is the riskiest thing. You can think of Canon or many other different, examples throughout the history. So what you need to do to convince these people is objectify the risk. So you need to quantify it in some way. And now I'm outlining the step that should actually always be done in drug development, and that is really stepping back and defining the problem, defining the context, and defining the frame of the problem. What's the purpose? What are we trying to achieve? What is our ultimate goal? How are we going to measure things, right? All this needs to be decided, and in pharmaceutical industry, it actually rarely is, whether it's a smaller company or larger, but these are steps that are followed in much more discipline in other industries that can put you in a position not only to convince, people that are afraid of taking new approaches, but also really making that you're taking an optimal approach. So once you frame the problem and you know what you're doing and how you're measuring things, then you should be specifying options, and these options should include one traditional design and should include a couple of completely different innovative approaches. And then you need to quantify that, and the best way to quantify that is really just to run simulations and get quantitative output for every scenario that you're considering. How do you measure? That depends on the value that you predefined. Is it minimizing the time, minimizing the cost, maximizing probability of success? Is it maximizing your return on investment? These are all things that need to be pre-specified, and simulations can give you answers to all these questions. Bayesian updating is clearly instrumental to this because it provides you with these probabilities, and it can be updated from stage to stage of development. Having an objective and optimal decision criteria is extremely important. So in order to have that, you need to prospectively define all your decisions. If you're defining decisions after the fact, after you see the data, you're not doing a favor to yourself. That's a well-known fact. And it has to be evidence-driven. It has to be data-driven. So here's one example of decision criteria. So in phase 2, oftentimes we have been defining the success using the p-value. If one dose is significant, then we proceed into phase 3. What does that p-value mean in the context of development? Well, nobody knows, but it looks good. That it looks okay at least. But why are you selecting that cutoff p-value? Why are you going with a certain sample size? All that is unclear. Now, if you produce a graph like this and you ask the question, well, if I keep on increasing the sample size in phase 2B, how much do I get? How much do I gain in terms of probability of success in the phase 3? Because that should be one of your most important questions. You know, what's the probability that my phase 3 is going to be successful? And generally, you will see that improvement with sample size will be relatively steep at the beginning and is going to level off. Now, with a graph like this, you're saying, okay, but if my sample size is too large, now the curve is flattening and I'm already spending so much and it's going to cost me time. Well, how do I decide? Well, a better way to go that I suggested many years ago, maybe, well, consider the expected net present value. Your financial decision makers are probably going to look into expected net present value anyway, and the expected net present value encompasses the cost and the time, you know, through expected revenues and the probability of success, because you can expect some revenues if you get onto the market, but you should multiply these revenues by probability of success in order to get the expected revenues. Well, the risk-adjusted revenues. So this is an example as the process of thinking that you should have as to what maybe, you know, the most meaningful outcome for you in a certain situation. I don't know how much more time I have left, but this is what the program should look. This is this is the approach that, in my opinion, companies should be taking, looking at their programs. So you look at your program comprehensively. So in this case, we're still talking one product, one indication. You design your trial, but you have different stages. They can be different phases, but they can be different. They can be stages between interim analysis, because you can have a number of interim analyses at each stage of development. You have internal information from your clinical trials, but you have external information that first influences your prior belief about success of your trial, but it changes over the time. And that information can be external data. It can be data from other clinical trials. It can be real data. It can be data from other clinical trials. It can be external data, like new things on the market that can be affecting your decision. Therefore, moving the hurdle that you need to pass, and as such affect your development. And finally, that this can be something that's happening within your company's portfolio that's going to have a direct impact on your program, and you need to keep on adjusting as you go your development. You need to keep on adjusting your development strategy and decision criteria based on all these parameters. But Bayesian updating is ultimately the tool that can help you digest all this information and keep on predicting the things in the future. I do want to cover portfolio level optimization just in a few slides. So one thing to consider is that at the program level or a trial level, we have two types of errors. One is to incorrectly continue development. The treatment is truly negative, so-called type 1 error or alpha error. Or you can be incorrectly stopping development of a good treatment, that's so-called power, you know, or type 2 error, or you're managing this to power of the trial. As you're increasing the size of your trial, you're improving both type 1, you're improving effects on both type 1 error and type 2 error in particular. However, when you start looking at the portfolio, there's a very important type of error that Bob Beckman and Kong Chen identified as a missed opportunity to invest into trials that might have identified good treatments. And it's hugely important not to commit this type of error. And the problem is that when you're increasing the sample size of individual trials, you are increasing this type of error. So you have a balance. You have a balance on which one do you want to focus. So this is how that works. I'm back to that curve of sample size versus the power. And at the beginning, that curve is much steeper, and then it flattens, and then in the end, it becomes a lot smaller. So for the last 1/3, or let's say 1/5 of the cost that you're investing, you're gaining very little in terms of power. So what you want to do at the portfolio level is actually remove the things, remove your investments that you have on the right-hand side to the investments that you have on the left-hand side as much as possible. You want to put your money where that buck is worth the most. So this is the concept of, and the reason why optimizing at the portfolio level is so important. If you think of, cardiovascular outcomes trial, $400 to $600 million. In last $100 million, they're just buying you 3% improvement in power. You may spend that on an indication that's much cheaper to develop another drug. You help your company, you help patients as well. This is the concept of optimization at the program level or at the portfolio level. So you're not optimizing at the individual program level, you're optimizing at the portfolio level. Platform trial. Now I have to talk about this. To me, this is the paradigm changer. This is what we will be seeing in the future. I have deliberately described it in a very similar way as the program level framework because there's similar aspects of this, but there are key differences, and that's if you look at the first arrow on the left, you're starting with multiple products or multiple indications or a whole matrix of products and indications. You have different stages because platform trials are inevitably going to be adaptive in their nature. So between each interim analysis, you can be making decisions. But decisions are not going to be optimal criteria now. There will be whether you should be closing. They will be focused on whether you should be closing certain treatments or certain indications because they're futile, they're not working, or maybe they should be taken outside of the platform into the next stage of the development, or is it time to add new arms to this platform? So it's a very it's a perpetual thing. So once you create this platform, which allows you to standardize, which allows you, if you really take care of the ways that you're treating the external data, allows you to include external data explicitly or implicitly into your decision making. This is to me the future of drug development because now you're getting a much broader picture. You can do this, let's say, for one therapeutic area where you have multiple products, but you also want to test them on multiple indications. It's all standardized, and it just keeps on going. And the more, as it proceeds further and further, it helps you more and more because now you have a richer database behind of that includes treatment arms that have already been completed. So to me, this is absolutely the way that we are going, the direction that we are going to. And this is a sub portfolio. So you will be making decisions, on a small portfolio kind of level. It's just a win-win approach in my mind. This is really just to illustrate that the platform is in a way a sub portfolio, and you can optimize at the platform level. But it's even better if you can conduct your optimization at the whole portfolio level that incorporates other products as well. And finally, something that I think I absolutely need to mention, and that is COVID-19. And why is that the case? Well, first, as COVID-19 emerged, there was a big question, how do you develop drugs? And there's a dilemma of scientific rigor versus the acute urgency for the treatment, and how much you can compromise. And let me clarify that platform trials are not a way of compromising, and multi-stage seamless trials are not a way of compromising. However, the question is, do you reduce the bar? Do you accept much higher levels of type 1 error, right? False negatives. Do you accept much smaller consideration of much smaller treatment effects? And do you accept the use of external data? And that would be very tempting because there's so much external data will be generated, very quickly for COVID-19. But the problem is, are they going to be harmonized? And are we creating a bias, by incorporation, and how to reduce the bias by incorporation of this external data, because you may be tempted to include the data that suits your cause the best. These are some dilemmas, but platform trial is not a dilemma. There are 3 ongoing platform trials. In fact, the very first treatment that is showing positive results, which is remdesivir, is actually coming from the NIH-sponsored ACTT trial. It's only 2 arms trial now, but the trial is open to adding other treatments. Solidarity, who sponsored trial, already started with 4 different treatments, and it also has the ability to add additional treatments as they become available or they are of interest to who. REMAP-CAP was actually an already existing global trial on community-acquired pneumonia, and it was open to adding new indications in a platform way. So, needless to say, COVID is already enrolling there in large numbers. So, in addition to this, FDA and EMA are considering are strongly recommending platform trials for development of COVID-19 treatments. So it's out there. All government and international agencies, government-sponsored agencies, and international agencies are supporting this approach. So I think it's just re-emphasizing the fact that the future may be in platform trials. Finally, the developers should be considering multi-stage seamless trials, such as seamless phase 2-3 trials. It's probably needless to test the dose separately, and that is actually what the first vaccine trial is intending to do. As much as I was reading the information, I'm a little puzzled as why that's the case. I'm not that familiar with developing vaccines, but I would just go with the seamless phase 2-3 trial. I know I'm not leaving that much time for questions, but I will definitely stop here, and thank you all for attending my presentation.
So before we move into the Q&A, we want to take a moment to remind you that EAST gives you easy access to a wide selection of trusted fixed and adaptive trial designs. EAST is used by nearly every major pharmaceutical company and the FDA. With a broad selection of popular designs in an easy-to-use format, we can help you quickly create and compare trial designs. Our company also offers a range of services, from staff that act as an extension of your team and consulting services for more cutting-edge projects to our new real-world analytics capabilities. We are here to help support your objectives. So now we are going to begin answering the questions submitted during today's presentation. So Zoran, our first question we have here: we only use fixed and group sequential designs. Are the other advanced designs you mentioned widely used?
Everything that I presented today has been used in most cases, even for approval of drugs. Group sequential design more than anything else. Naturally, phase 2 adaptive designs in phase 1 oncology, adaptive designs a lot, particularly by large pharma, a little bit less by smaller biotechs. But these are other common approaches. As I said, unblinded sample size reassessment is becoming a very common design. Seamless phase 2-3 has been used and is responsible for multiple approvals by the FDA based on a single, seamless phase 2-3 trial. The subpopulations, adaptive subpopulation selection has definitely been used, maybe to a little lesser extent than others in the phase 3. Nevertheless, it's something that the FDA would really like to see. Master protocols, large pharma are really stepping into this area. And actually, I have seen biotechs who of a size that can create the master protocols and platforms are actually going more and more into this area. So this is not a new thing anymore, but it needs a lot more application, in my opinion.
Thank you, Zoran. And our next question: could you expand on the vision of platform trials? How will they change drug development?
Yes, so I might have done that already in that last slide, but I think just the advantages that one gets with a platform trial. You start with something, you create a platform, you standardize your processes in that platform, and then you start adding all your products. So if it's a therapeutic area, let's say, you just start adding anything related to that therapeutic area, all indications, all products that you have. And now you start creating one standard database, and you keep on building on that. And the more and more that you are adding to this, the more and more, as I said, you're benefiting from this data. Another very important thing that you should be doing at this time is think of databases and how to use, how to take advantage of really the rich information that you have in external databases, whether from registries or from other clinical trials that you may get access to, or any other information that can support your decision. So that platform will be sitting on this real world evidence, and would be building, you know, clinical trials on the top of that. And you can be using, I definitely think you would be using these Bayesian criteria, you would be using this background data to create priors and keep on updating your priors to make decisions in a more and more efficient way. And everything would be done, you know, in a very standardized way, because that will be regulated by master protocols. So that's my vision. I see the industry going in that direction, but I definitely see government agencies wanting to see more and more and more of this. So that's kind of what I would like to say in addition to what I said already said during the presentation.
So we just have one last slide here. So this slide here shows the schedule of upcoming webinars as part of this new series that Cytel is launching on complex innovative trial designs. So you'll see that our next webinar by Professor Jennison is on Wednesday, June 3rd at 11 a.m. Eastern Time, and we'll be covering group sequential designs and sample size re-estimation. So thank you, Zoran, and thank you to everyone for attending today's webinar on innovative drug development at a glance. On behalf of Cytel, thank you for joining us today, and I hope that you have a great rest of your day.